Welcome everybody to this session. This is the fourth installment of our Grant Writers Toolkit series. Dr. Betsy Bateman from Brigham is co-moderating this session with me today. On the agenda will be a brief didactic on navigating the NIH grant submission and review process, a mock study section discussion, and dedicated time for a Q&A at the end. We anticipate the session will be about an hour, maybe a little bit less. If you do have questions that come up during this discussion, feel free to put them in the chat, and we can either address them on the fly or during the Q&A at the end. I wanted to thank our formidable group of panelists today for this session. They include Dr. Karen Ridge, professor at Northwestern, Dr. Chen, who's the professor and division chief at Cedars-Sinai, as well as interim department chair. Dr. Yeligar is associate professor at Emory, as well as Dr. Kolls, who's the professor of medical pediatrics and the John Deming Endowed Chair at Tulane. Thank you again for taking the time to lend your expertise and your perspective today to all of us. And so, as I mentioned, we'll start with a short talk about, what happens to your grant from submission to review. In years past, this portion of the session was done by scientific review officers from the NIH. Unfortunately, as you may be aware, the NIH SROs are still under policy-related communications bans and are unable to participate today. My understanding is that they're still available for one-on-one meetings if you do have questions related to upcoming grant applications. Whether that'll change anytime soon, I can't say. I've been asked to stress that the following slides should not be considered on behalf of the NIH. They contain largely publicly available information, and unfortunately, I don't have particular insight or insider knowledge about policy changes in the works and how they would affect the submission review process or the content of this talk. So in the next 10 or 11 slides or so, I'll touch briefly on each of these topics. First, what happens to a grant once it's submitted. Second, how reviewers are chosen. 3, the NIH scoring system and the new simplified review framework that went live this year. Who to talk to about what at the NIH, and some key points about early stage investigator status and the benefits that come with that. So what happens when your applications once it's received by the NIH, and how is it assigned? So all grant applications go to the Division of Receipt and Referral. That's within the Center of Scientific Review, or CSR. After receiving applications, DRR checks the applications for completeness. It determines the area of research and assigns it to a specific NIH institute or center. DRR also assigns applications to specific study sections, also known as scientific review groups, that have the specific expertise to evaluate its scientific and technical merit. The NIH uses a dual peer review system that's intended to promote a review process that strives to be fair, equitable, timely, and free from bias. This first level of study review is carried out by the Scientific Review Group, composed mostly of non-federal scientists with specific relevant expertise. Until recently, about 20% of grants were reviewed in study sections administered by individual institutes and centers. But as of March of 2025, CSR now coordinates the first level review of all grant applications, R&D contracts, and cooperative agreements across the NIH. The intention of this centralization effort was to reduce costs and to mitigate bias by completely separating the review and funding components of NIH. The second level review is performed by the Institute or Center National Advisory Council. These are composed of scientific and public representatives chosen for their expertise, interest, and activities in matters related to health and disease. And only applications recommended for approval by both the Scientific Review Group and the Advisory Council can be recommended for funding. And ultimately, final funding decisions are made by the NIH Institute or Center Director. In initial peer review meetings, each notice of funding opportunity, also known as NOFOs, specifies the review criteria and considerations that will be used in the evaluation of those applications. Each scientific review group is led by a scientific review officer, or SRO, and an SRO is essentially an extramural staff scientist and designated federal official that's responsible for ensuring that each application receives an objective and fair initial peer review, and that all applicable laws, regulations, and policies are followed. Each scientific review group is composed of a chair that serves as a moderator for the discussion of applications, peer reviewers, and other NIH staff. An initial peer review evaluates applications for scientific and technical merit of the work proposed, its overall impact, and appropriate justification for human subjects and vertebrate animal studies. And so how are reviewers chosen for particular grants? And so, as you know, the NIH peer review system relies heavily on the participation and expertise of accomplished, broad thinking, and creative scientists. Peer reviewers, by and large, are non-federal extramural scientists that are active and recognized authorities in their field. There's also an emphasis on a balanced representation of reviewers, and this could include diversity in geographic distribution, institution, gender, race, and ethnicity. Fairness and objectivity, as you might imagine, are crucial for reviewers, and conflicts of interest are therefore identified and managed. So reviewers can't be in a study section reviewing a particular application if their name is on that application in a major professional role, if they're a member of the NIH Council, or if the reviewer or a close family member would receive direct financial benefit. And after identifying potential reviewers, other information is further used to determine eligibility. And these will include things like extramural funding history, publication history, and prior review experience. And for each application, 3 or more reviewers are assigned to provide an in-depth assessment, written critiques, and criterion scores. So each NOFO is going to provide information about the application as well as their specific review criteria. So check out section 5 in each of these NOFOs that you're looking at in the future. The application review and scored review criteria can differ depending on the grant mechanism. And there are 3 general categories for all applications, and these are unchanged in the simplified framework we're going to discuss shortly. Applications receive an impact score determined by these review criteria, as well as an overall impact score to reflect the reviewer's assessment of the likelihood for that project to exert a sustained and powerful influence on the research fields involved. The first category of scored review criteria require criterion scores on a scale of 1 to 9, and these scores play a role in determining its overall impact score. There's additional review criteria, such as human subjects protections, that don't necessarily require scoring, but will influence the overall impact score. And there are additional things, such as the budget, sort of period of support, that don't require scores and aren't supposed to have an effect on the overall impact score. Criteria, for those of you who may not be aware, are scored on a scale from 1 to 9 in whole numbers, with 1 corresponding to exceptional and 9 corresponding to poor. The NIH expects a reasonably normal distribution, namely scores of 1 to 9 are going to be used a lot less frequently than the others. And after review of the application, all members will provide their final scores privately. The overall impact score for each discussed application is calculated by taking the mean of the reviewer scores, multiplying it by 10, and that's what's reported on the summary statement. So to talk a little bit about the simplified review framework, this was developed in response to sort of repeated concerns about the increasing complexity and administrative burden of review criteria over the years. In response, the NIH developed the simplified review framework, which went live for all applications submitted on or after January 25th, 2025. And so the ultimate purpose of this is that the NIH wanted to ensure that peer reviewers could really better focus on the key questions. Should the proposed research and can the proposed research be conducted? And ultimately to mitigate the effect of reputation of the PI and institution. So these criteria that are put forward were meant to contextualize the review to be more clearly within the framework of the research being proposed, as opposed to having an individual or institution's reputation carrying it forward, even if there were methodological flaws. And this framework reduces the reviewer burden by moving unscored review criteria, particularly those about policy compliance, and shifting that onto NIH staff to review instead. So in the past, this sort of kind of a before and after infographic. In the past, there were 5 criteria, and now the idea is that they're shifted and kind of recombined 3 factors. You'll see on the right panel in green, factor 1 emphasizes the importance of the research. Factor 2 emphasizes rigor and feasibility. And factor 3 considers expertise and available resources for the grant. So factors 1 and 2 are going to be scored as have been previously done and given criterion scores of 1 through 9. Factor 3, which covers, again, environment and the investigator, will not be given a criterion score. The reviewers are prompted to say whether the expertise and resources are appropriate or not appropriate. And if they're not appropriate, they're prompted to give comments about why it's not. And again, the idea here is to avoid reputation carrying a grant in the way that it maybe shouldn't. So to kind of summarize these new factors as one-liners, factor 1 really asks whether the research should be done. Factor 2 asks, can it be done? And factor 3 asks whether the resources are appropriate. The factors that reviewers are being asked to consider are condensing as well. So the additional review criteria that can affect the overall score still include things like human subjects protections, vertebrate animal protections, biohazards, and considerations regarding resubmissions and renewals and revisions. Other criteria, such as the study timeline and the inclusion of women, minorities, and children, are being sort of shifted and incorporated into factor two for those relevant studies. For other review considerations, reviewers really will only have to comment on the authentication of key biological and chemical resources, as well as the budget. And other criteria, such as select agent research, resource sharing plans, these are now being shifted to NIH staff. So one thing that kind of confused me in the past, and to be honest, can occasionally confuse me now, is when you submit a grant or before you think about submitting a grant, who are you supposed to talk to at NIH? When are you supposed to talk to them, and what are you supposed to talk to them about? Like, what's the difference between a PO and an SRO and all these things? So hopefully I can sort of elucidate that a little bit here. And broadly speaking, there's really three kinds of NIH staff that you're going to interface during a grant life cycle. The first is program officers to the left, and you can reach out to these folks before applying about exploring the scientific alignment for your proposal, whether that NOFO is an appropriate funding mechanism, or if you have questions about particular review criteria. After your grant is reviewed and you receive a summary statement, the program officer assigned is someone that you can go to talk to about resubmitting, or if you have any particular questions about the summary statement. Program officers are also the ones that read progress reports for awarded grants, so they can be resources after the award, and they might get in touch unilaterally with questions regarding moving goals. So as an applicant, sort of in this middle column, your interactions with scientific review officers are going to be pretty narrow. Really, the only time you'd be interacting with an SRO is after your application is submitted, but prior to review. And this is mostly to discuss additional documentation that you might want to submit, post-submission materials, sort of updates at that 1 month mark, or if you have any concerns about conflict of interest regarding people in the scientific review group. And on the right, grant management officers, specialists, these are the folks that are in charge of budgetary policy really at any stage during the grant cycle. So you can talk to them before or after about using funds in a certain way, reallocating funds or reassigning funds if you're moving institutions, things like carryover, if you're getting into the grant, if you have a lot of money left over, these kinds of things. And so the last sort of major topic that I want to discuss that's going to be relevant for folks in the webinar here is early stage investigator status. So what is this and why do you care? And so this status is sort of specific to the NIH, and that's someone who's completed their terminal degree or postgraduate clinical training within 10 years and has not yet been awarded a substantial competitive NIH research grant, namely an R01 for which you're the PI. This status is actually automatically determined based on what you put in the sort of ERA Commons profile regarding training, so just make sure that's up to date and accurate. This status can be extended for particular circumstances, most commonly childbirth, but other reasons can be considered on a case-to-case basis. And this really can be anything that takes you away from the bench for a significant period of time. And so why do you care? So the purpose of the ESI designation is to create greater parity in success rates between early stage investigators and established ones. So peer reviewers, at least, in a platonic ideal, should be looking at ESIs a little bit differently, since it's not necessarily realistic to hold people who haven't been in the field as long to the same standards regarding track record and publication and achievement. ESI status prompts study section to look more at potential rather than at what investigators already accomplished. And from a practical perspective, where feasible within a study section, scientific review officers will kind of group ESI applications into their own bunch to kind of keep reviewers tuned to that sort of ESI wavelength. So for the purposes of funding, the NIH will prioritize ESI R01 applications with meritorious scores for funding. Some institutes may actually set separate success rate targets specifically for ESIs. What I can say based on information shared at previous iterations of this session is that NHLBI historically tends to give a 10% bump to ESIs, as well as a 5% bump to sort of first renewal applications or second R01 applications after an awarded ESI application. So definitely worth looking into and making sure that your training history and everything in ERA Commons is up to date and accurate. And if you have an extenuating circumstance that's sort of taking you away from the bench for a while, applying for those sort of extensions and stuff would be helpful. So the last thing that I want to briefly mention that may be relevant to us is this early career reviewer program. So this is an opportunity to participate as a reviewer on a study section as an early career scientist. The qualifications, as I've listed here, include 1 year of full-time faculty at the assistant professor level or equivalent. You've had some senior author publications. You haven't held an R01 or equivalent, but you have submitted a grant in a PI role. This could be an R21, a K, so on and so forth. Previous panelists who have participated in this program have really found it invaluable. You've generally assigned fewer grants to review, which makes it kind of less intimidating, and sort of being in the room to listen to these conversations sort of helps improve their grantsmanship. So that is all I have regarding the SRO portion, the sort of grant lifecycle portion. Unless there are any particular questions at the moment, what I can do at this moment, I don't see anything in the chat. What I'll do is really just start the next and sort of meat of the session, which is our mock study section. You should have gotten an email regarding the application materials. Having them is not necessary to benefit from this, but if you don't have those materials, I can ask Samantha to sort of email them out once again. Just message me or message Samantha in the chat. So with that, I will kind of shut up a little bit, and then I believe Dr. Ridge is going to be serving as our sort of chair of this session, so I will kick it to her.
All right, Chris, thank you so much. So we're going to begin the mock study section, and just as a reminder, we're going to ask the 3 reviewers to begin with their initial scores. They'll summarize their critique, the strengths and the weaknesses, with a focus on whether or not it should be done or can it be done, and if the necessary resources and expertise are available to complete. Reviewer 1 will have about 7 minutes, reviewer 2 about 5 minutes, and reviewer 3 about 3 minutes, and I'll call out if you go significantly over the time. So with that, could I ask the 3 reviewers for their initial scores, please? Dr. Yeligar.
1.
Thank you. Dr. Chen.
5.
Thank you. And Dr. Kolls.
3.
Fantastic. All right, Samantha, please take it away.
Okay, so this is a new application by an early stage investigator who's proposing to study fibroblast and corpse interactions. So corpse is defined as cell debris or dead cells that occur after lung injury, and how this fibroblast corpse interactions might play a role in lung repair after injury. They're proposing to use a bleomycin-induced injury model, and they have some preliminary data to support this interaction between fibroblasts and corpse cells, namely through a phosphatidylserine dependent AXL signaling. So this promotes sort of fibroblast activation in terms of interaction with another molecule, EIF5A, which is needed to promote lung repair. So this interaction kind of increases collagen synthesis and allows for more repair to be done after injury. So they're proposing to use in vivo lung slices in some human primary mouse and human fibroblasts, as well as fibroblast targeted knockout mice to do these studies. And I was really excited about this application because there hasn't been a lot of focus on the role of fibroblasts in lung repair. So I thought it was a very understudied area, and the way that they're proposing to use these different models is very translational. And so I thought it was very innovative, just conceptually in trying to understand how fibroblasts can actually work within lung repair after injury by looking at how this recognition of these corpses actually happen, what's actually needed after the fact, and how repair happens within the cell as a result. So the group has been very successful in the past, and there are resources available to be able to look at the questions that they're asking. As I mentioned, they had a lot of preliminary support for their scientific premise, and so they're proposing that this AXL signaling is needed to promote fibroblast recognition of corpse phosphatidylserine, and that in turn increases this fibroblast hyposine EIFA like interaction, and that's needed for collagen protein and increased fibroblast polyamines, which is needed for repair. They've proposed using male and female cohorts. There's a lot of rigorous design because there's blinding of groups. There's more than 2 independent experiments for all studies. I thought that the potential pitfalls and alternative considerations were very well described for each study and not even just each aim. The only issues I kind of had from a grantsmanship perspective were some of the preliminary data figures were too small to easily read the axes. And I thought that since they were proposing to have AXL knockout animals, that doing sort of a gain of function side of things to be a complementary assay would be beneficial to sort of promote what this as a critical mediator to the interactions between fibroblasts and corpses.
Okay, thank you. Reviewer 2?
All right. Well, I was a little less excited about this application for multiple reasons. First of all, the investigator has worked largely in macrophage biology and is switching over to fibroblasts. So I don't know if this person actually has the expertise to do this kind of work that's solely on fibroblasts. And I think it kind of panned out. And if you look at the grant, it's very largely focused on an in vitro phenomena that I don't know they're going to adequately study in vivo. And so all the work that they're going to do in vitro, looking at hyposination and polyamines and everything else, could just be an in vitro phenomena. And none of that is really going to be explained in vivo. I think that they really need more preliminary data to support their aims. And particularly in aim 3, they have all these mice lines they want to do, and they don't have any data supporting those animal studies. The other issue is that they've really locked in on this idea that it's an interaction between the fibroblasts and these apoptotic corpses. But as these cells die, they start releasing things. And so how do they know that it's actually a true cell-cell interaction and it's not through some secreted mediator that is actually regulating the effect? And so I think the studies will lack in the ability to lead to anything translatable in the future. One of the final issues I had with the grant was that it largely focused on repair. The hypothesis is all about fibroblasts recognizing corpses for repair, but all their endpoints was on fibrosis. And so that's not necessarily the same thing. And so I thought they were very misguided in that.
Okay, thank you. All right, Jade, can you bring us together?
Yeah, so I was in between with the 3. I think, conceptually it's an innovative hypothesis. They have some really novel tools, and including the AXL knockout and also additional ATSF1 knockout, which is a rate-limiting enzyme that controls the putrescine generation and polyamine synthesis. My major concern was a lot of the phosphatidylserine exposed cells were immortalized cells like Jurkat cells or immortalized epithelial cells as opposed to using primary cells. And particularly during acute lung injury, there would be a lot of apoptotic neutrophils, and so how these fibroblasts respond to apoptotic neutrophils, I think, would be really important. I did like the organization of the grant, the way the PI kind of summarized what each aim was going to do, and then had a second section like where drilling out the experiments. There was some weakness in the statistical approach. The PI said he's going to use a student's t-test, but obviously that assumes that the data is Gaussian, and maybe the data is not Gaussian, so he could have a non-parametric analysis of the data addition. And then when I saw the list of mice, I always get nervous when I see a B6129 SF2/J mouse, which means that that mouse was originally made on a 129 ES cell and then crossed to B6, and they didn't really specify how many generations. So, and as in caspase 111, and so which obviously can control necroptosis and pyroptosis, and so it'd be important to make sure that those reagents were fully back crossed to B6 to have congenital controls. And then lastly, a lot of the readout is collagen synthesis, right? And there's a whole sub-aim about looking at transcription, and transcription is assayed at one time point by qPCR. Well, that doesn't measure transcription. That measures the amount of mRNA present at that one time. To measure transcription, one would have to do a nuclear run-on type assay and also potentially assess mRNA half-life to see if that's going to. So I thought, from a little bit of weakness in the molecular biology approach and in that later AIM 1. And then I agree a little bit with Peter. They do have some data that condition-mediated does not confer the phenotype, suggesting that fibroblast apoptotic cell contact is required. But the real critical experiments are AIM 3. So AIM 1 and 2 could show this elegant in vitro phenomenon, and without some additional data showing that this occurs in vivo, I think does impact the overall significance of the project without a little bit more preliminary data. So I agree with Dr. Chen on that point.
Okay, thank you. Before we open this up for discussion, can you let me know if they've addressed sex as a biological variable, and human subjects, please, invertebrate animals, any issues?
No issues on vertebrate animals. They talk about sex as a biological variable, and there's no human subjects.
Okay, then this application is open for discussion. So I have a quick question. I was a little bit concerned that their endpoints were seemed to be over-focused on collagen deposition, but there was some pretty significant changes in permeability as assessed by albumin in the bronchoalveolar lavage fluid, and it didn't seem like how the hypothesis was going to address that. And perhaps I missed it because obviously I only gave this a cursory review, and so if any of the reviewers could address that.
Well, I mean, that was one of the main reasons why I didn't like the grant, and that they weren't really connecting the dots of what they said they want to do as far as their hypothesis and what they were doing. And so I don't think they adequately addressed this concept of barrier function in their models.
I think the way I was reading it and why I was so enthusiastic about it was that I was reading the hypothesis in this area of study as being a contributing factor and an area that has really been understudied in the field. Not necessarily that this is the only path or it's like maybe even the most critical. I think I was taking it as there needs to be a starting point. And so for me, that's why I was so enthusiastic to see this, because I thought it could be a good starting point.
Yeah, I think most of the albumin work was in the context of so they're doing two models of lung injury, LPS and pleo. And so it was in the context of the LPS model. And they do have an AIM 3 as an in vivo that they're going to measure total protein and albumin after LPS inoculation into this conditional, either the actual flox mice or the ODC1 flox mice that's crossed to PDGFR3 to knock it out those pathways specifically in fibroblasts.
Are there any other questions from the panel? Well,
I just wanted to, sorry, I wanted to make one comment on what, on one of the weaknesses that Peter mentioned, where the PI is more focused on macrophage biology in their past. There is a co-investigator that is on the investigative team that seems to have some effort to, that does have a background on fibroblasts and lung repair and a little bit more expertise that I think could help this early stage investigator with some of those questions.
Okay, thank you. I heard a couple of comments on how they didn't have sufficient preliminary data to support AIM-3, but I just want to remind the panel that this is an early stage investigator and that extensive preliminary data is not required, but it does need to be there in order to establish feasibility, which is also an important new factor of evaluation. So based on what you've read, can this application, can the proposed research be conducted by the group being reviewed right now?
Yeah, I guess you're right. I was probably overly harsh there, and they can cross the mice, and so I guess they can probably get the experiments done.
Dr. Kolls, do you agree?
Yeah. I mean, we have ODC1 flocks in my lab, and they don't have any issues. But I'm not that familiar with the axle knockout, but they've been around for a while. So I think one issue though is still the significance issue, right? So if all your preliminary data is wrapped around just in vitro phenomenon, does that give me assurance that the research has enough significance? So so having a little bit of in vivo data that I think would help that issue.
Okay, I have just two last questions. There's two different subunits of this EIFA 1 and 2. Did they address both of those? Because I only saw an A1 during my quick look through the grant. That's question one. And then I didn't see anywhere that they were really looking at what the impact of this hypocene pathway was on mitochondrial function, which has been known to really have a pretty significant impact.
Samantha, you want to address that or?
I didn't see about the different subunits of the 5A. I think while it does, hypocene does have an effect on mitochondrial function, that was kind of outside the scope, I felt, of the current studies that were proposed. So even though that's important and maybe, could potentially be considered as another consideration, I just didn't really think it fit with the current proposal.
Okay. All right. Well, we're right at the 15-minute mark. That was a pretty good discussion. Thank you, everybody. So right now, just as a heads up to the panel, chairs no longer summarize the discussion anymore. So that part has been eliminated, which makes chairing a lot easier. So we're just at the point right now that we're going to go back and ask our three primary reviewers for their final scores. So Samantha.
2.
Okay. Peter.
I'll come to a 4.
Okay. And Jay.
Yeah, I'll stay at a 3.
So the range is between 2 to 4. If there's anybody voting outside that range, please raise your hand. We'll make a note of it. And before we move on, can I have any comments from the three primary reviewers on resource sharing, data management, budget, select agents, and authentication? Any issues?
No issues.
Okay, then that completes the review of this application, and I'll turn it back over to Chris.
Thank you, everybody. That was really great. Before we open it up for the sort of Q&A, again, folks, you're welcome to raise your hand, put anything in the chat. before we kind of jump into it, now that we're sort of out of the mock study section, was there anything that you all really wanted to kind of highlight that went well or went badly about this grant as you were reviewing it?
I think the grant lacked a central figure that would have been critical for the non panelists that get to equally score this grant. And you might only have a minute or two to quickly look at it before the discussion happens. And those primary figures, those schematic figures, are really key to guiding reviewers who are scoring your grant in the same weighted way that the three primary reviewers are.
Yeah, I agree with that. I think most, grants will put a schematic in the specific aims. And you can really, if it's done right, you can understand the grant very quickly by just looking at that schematic. And so for all the people on the panel that are just skimming over things, you want to give, you want to force feed the information to them as easy as possible. And I always say, just treat reviewers as they're stupid. And make sure that you just make it as easy as possible for them, because they get irritated if they have to think about it.
Yeah, I reviewed this like I would if I was reviewing for a study section. So, even if I had time on a day, I made sure I did it at the end of a work day. So I had that tired mindset because I will say, so Karen's question about the subunits, I had to actually go through and look really quick when we were talking, because it wasn't something I remembered from a week ago when I read this. And so, since I couldn't quickly find the information, I had to say that I didn't see it. But I think that keep it simple, concise is really important.
Yeah, I agree with the overall figure that Karen mentioned. There is some minor issues, like the PI says that the corpse binding is really the necessary trigger to get the fibroblasts to turn on this pathway, but they never really. And they mentioned some fibroblasts engulf, right? Bind and engulf, right? And some just bind. But so they mentioned that they see this upregulation of collagen in the fibroblasts that bind, but they don't compare that to the ones that bound and engulfed.
Yeah.
So there is there's and that they use the term necessary, which I think in some cases they may just have sufficiency data, not true necessity. So I think it's just going to be, it's important, I think, not to overstate your data.
I totally agree. I was supposed to be the nice person, but totally agree. Things were overstated.
Thank you for playing the role of the nice person.
I also think those types of comments are often tempered when you're reading an ESI grant as opposed to an established investigator grant. So
I agree.
I think that if this was an established investigator grant, the scores would have been, in reality and in this mock session, a little bit different than what we're seeing. And I do think reviewers do take that into account on whether this is an ESI grant or not, which is important because we all started young in this business and we all, make mistakes and we grow from those. And so you have to have a little bit of grace when reviewing ESI applications.
So I had one technical question maybe for the group reading this grant, was that they're using the PGF-FR alpha Cree mice, but then they're using it crossing to an AI-14 mice so they can specifically target the fibroblasts that showed recombination. Is it because that Cree is just not, has some degree of chimeric recombination? Is that, I don't work with that mouse, so is it known to have some inefficient recombination?
Yeah.
Yeah.
So one thing to do, but one thing I like usually say yes to study section is because I learned something.
Yeah, the other criticism I would have given this is the endpoints, because a lot of that remodeling occurs much later than I think some of the endpoints that they were looking at, particularly in the LPS. Like you could get barrier restoration, but the extracellular matrix remodeling can occur, days to weeks after that initial injury. So I'll be curious when this paper comes out, whether or not they saw it at the early time points or not, because my bias would have been that we saw it much later. But, often wrong, never in doubt. We'll see what the data says, right?
Any other burning questions from the audience?
I actually have a question for Dr. Yeligar. You had mentioned that you tried to review the grant as if you were actually reviewing the grant, like at the end of the workday when you're feeling tired. I was wondering if the panelists could talk a little bit about that, like in what context you often review grants, and when you look at a grant, how you review it, like what sections do you start with, kind of your personal process.
I usually just kind of go straight from the beginning all the way through, honestly. I just follow how it's kind of laid out, getting a sense of what it's about, and then the investigators, and then just going into the meat of the grant. But the majority of the time is spent on the specific aims and the actual studies. I don't spend that much time looking at facilities and equipment and even the bio sketches. I just kind of really go straight to the grant and read that, because that's where the it sways in my mind. It's usually not something's missing the bio sketch, or they didn't do this, or they need to do this, publish this. That's not what gets me. Everything else kind of falls in line after I figure out if the grant is good or not. It's kind of how I approach it.
Yeah, and I typically, sometimes I'll carve out some time on my Outlook calendar to review grants during the week. But oftentimes it's, on a weekend, and if it's winter, maybe a little fire going. I get you in a chill mood, read some science.
I always tell my trainees, you have to write your grant so whoever's reading it will still like it after they had a fight with their spouse because they didn't take care of dinner. And the kids are trying to learn their math homework at the kitchen table while you're reviewing their grant. And if you can write a grant that's clear enough that I can understand what you're trying to do with that chaos, then you have a great hope of having a good score. And that sounds terrible, but there's a lot of truth to that. And then the other thing is that if it's a renewal and I didn't review it in the first cycle, so it's a resubmission rather, I don't read the critiques until I've read the grant. So I try to say, like, let's give this PI the benefit of the doubt that perhaps the previous reviewers didn't understand something or misinterpret it, and then I'll go back and see, were they responsive and did I agree with those critiques? But I'm not sure everybody does that.
Yeah, I agree with you there, Karen. I kind of generally do that also, is read the grant and then try to give them the benefit of the doubt. Problem is sometimes you find things that weren't brought up in the first review, and then trying to bring all that together. And I think again, I agree. You have to write this grant so that people can understand it and be very concise, to the point. I always tell people critical points that you're trying to bring across in the grant, just keep saying it over and over, just to kind of make sure that the reviewer knows it, because they're going to be skimming it pretty quickly. And so that if they miss a certain point, then that is important for the grant. You see this all the time. They're like, people will bring up certain things about a grant, and then they're like, well, they actually wrote it right here. And they're like, oh, okay, but it doesn't change my mind. And so you don't want to lose the reviewer early on.
Yeah, I agree. Some degree of repetition is good. One thing that this PI did was, at the end of this aims page has a series of deliverables of what this grant's going to deliver. But, at the end of the approach, the last page doesn't close the loop again. So sometimes it's helpful to have a little couple sentences at the end to say, if we accomplish this aims, this is how we'll advance the field. So kind of have a 360 kind of approach.
Well, one advice I'd give to ESIs when they're writing grants is that you should always try to find people to read it. And they don't have to actually be people right in line with your research. You actually want people that aren't in line, because you want people to be able to understand your grant very quickly and easily. And
I agree completely.
you're going to have people that are reviewing the grant that are not they don't understand your stuff that well, but they are scientists and they understand how it should be done.
That's great advice. I'll have two other comments to that. So one, what we do here at Northwestern in our group is we have a read out loud day. So we literally read the entire grant from summary all the way to the final page out loud, because you'd be amazed how many grammatical errors you catch, or how many incomplete thoughts that you could end up having. So you have to also give a day for that as well. And then in response to one of the questions in the chat, it was any tips on how to improve interpretation, potential pitfalls, and alternative strategies. It feels like how to read those sections in the grant. I think those are some of the most important sections of a grant, because like some of the comments that were brought up were like, well, is AXL the only receptor that's actually going to be involved? Like, is MERTK going to be involved? And you could put that in, if you were to say, I'm going to look at all the potential phosphoserine receptors that could be activated in this pathway, then you would be charged with having an overambitious or unfocused grant. But in your interpretation, pitfalls, and alternative strategies, you could say, there's a possibility that AXL isn't the key receptor, and so we might have to explore other ones, and based on our RNA-seq data, we'll prioritize them, for example. So those alternative strategies are really key, because it makes the reviewer appreciate that you've really thought through all the same types of potential weaknesses and strengths of your own rigor of your research. So I can't overemphasize enough how important those sections are. And I'll let others comment on that as well.
And I would like to add, if you think of an alternative strategy that is really exciting to you, it's going to be really exciting to the reviewer. And so maybe consider just adding it as a sub-aim potentially, because, bringing something up that you have a lot of knowledge about, or you think is very important in that section, automatically the reviewer is like, yeah, you should do that. I would love to see that. I'm wondering about that too. So, when you're thinking through these things, think about how to improve your proposal as well.
Well, what I would say though for ESI grants is you don't want to make it too complicated. I always tell ESIs, you don't need them to love you, you just don't want them to hate you. Because to get an ESI grant funded, you don't need to have threes, you just need fours. And so if you try to get too out there and novel and to show that you really are doing some cool stuff, then you just open yourself up with lots of targets.
Yeah, so
That's why you get feedback.
Yeah. So this PI proposed the MERTK pathway if the AXL pathway, turned out to be a dud. And so the first thing I did when I saw that was I went to untabular mirrors and looked at lung fibroblasts and made sure that they expressed the MERTK, which they do. So so that was a good alternative. In my mind, that was a good alternative because of the fact that they share some signaling. So
Yeah, I think, Jay, I do the same thing. I'm always checking, like, does their gene of interest actually, is it expressed in the models that they're using? And is it expressed in human tissue as well? Because if it's not, I'm like, well, why are we studying something that, doesn't have the potential to go forward, right?
Yeah, you see it on study section too. People will be like, oh, I got on this atlas and it doesn't express this gene. And so that should be your first thing you do is make sure your genes are expressed where you're saying it is.
although the caveat, it depends on the depth of the single cell, right? How, so
No, I know. I get it.
but so that's why I always look at bulk lung data as well as single cell data.
But you see it all the time on study section. People are
Yeah.
just sitting there searching their atlas, their favorite
Yeah,
atlas
exactly.
for stuff.
I was hoping to actually ask a follow-up question. Dr. Chen, you mentioned this line of you don't have to get them to love you, you just have to let them not hate you as an ESI. Sometimes I sort of feel this kind of like tension as an early stage person between this sort of like innovation and feasibility, right? Like a grant has to be both like pioneering, but also be practical, likely to succeed. And I was wondering if you guys had any thoughts about kind of how to balance these competing priorities, or if you can think of instances or examples where it was done well or poorly or anything like this.
Well, it's kind of hard to give general advice on that until you actually see the grant. I think that for me, it's always you have to have an innovative question. What you're studying has to be very exciting and new, and then the approach has to be innovative enough. There's in general, our approaches are not, novel. There's no technical innovation in our approaches, although we always write it down as technical innovation. And so you just kind of use your standard approaches, but you have to just ask good questions. And I think that's what it comes down to.
I think there's a question about statistics in the grant, and the PI put the statistical section in the rigor of the proposed research paragraph, and addressed both when to use two sample tests versus more than two sample tests like ANOVA, but also that's also where the PI put their power analysis for the number of mice per group. But when you put a power analysis, it should be powered around whatever your primary outcome measure is. So if it's lung leak or if it's collagen expression, you should, because you need a primary outcome measure to do your power analysis. And obviously for human subjects, you're going to have a much more sophisticated statistical, and because then you got to control for many covariates and other things. So, my experience is that with large human subjects grants, I mean, you almost have to have a biostatistician as one of your key personnel, right? So,
Jay, I think that's a really good point. Don't be afraid to have a lot of consultants. So we really tell all of our ESIs, go get the core director of the flow cytometry core to write you a letter. Go get the director of the sequencing core to write you a letter. I mean, the more consultants you have that support these techniques that may not be routine in your lab yet because you are an ESI, I think it further strengthens your application, and letting the reviewers know that if you hit a problem, that you've got experts at your fingertips ready to support you in whatever issues that might come.
So we are reaching the top of the hour here. Are there any sort of final burning questions or parting wisdom from our panelists?
Start early.
Yes, definitely. Well, thank you everybody for attending, and thank you in particular to our panelists for taking the time and having a great discussion like this. It's very much appreciated, and hoping to connect with you all at ATS going forward. Thank you, everybody.
Thanks.